Awesome.
Fabulous.
Well, thank you so much for that introduction, and thanks everyone for being here. I'm really excited to learn more about AgBioData, and then also to talk a little bit about some of the work that I've done in looking at crop wild relatives in herbaria. So this is also kind of in the introduction, but I just wanted to kind of introduce who I am and why I am where I'm at. But I did my master's and my undergrad at San Diego State University. And during my master's, I studied the systematics and biogeography of the genus Cryptantha and the Boraginaceae. So got to do some field work in pretty cool places like the Andes to find these plants, which hadn't been collected in many decades. So that was really introduced me to how fun it is to go out and try to find these different plants. Then for my PhD, I was at the University of Missouri and studied the domestication history of Brassica oleracea. And so we grew a lot of these in the greenhouse, but I was really interested in from some of the results of that work in trying to understand these wild plants that looked more cultivated than wild, and those likely are of feral origin. So, kind of continuing to be interested in these feral plants and then also these wild brassicas, which then leads me to where I am currently at the Florida Museum of Natural History at the University of Florida, where I've been working partly a few different ways. So one way with using this AgBioData to look at how this digitized herbaria reveal opportunities for and risks for crop wild relatives, especially when we look at climate opportunities. And then also, so this is collecting Brassica rupestris in Sicily, and then also with USDA, looking in more detail at the origins of feral brassicas. And so I know I don't have to convince this group. Let me just hide this. That there are tons of climate hazards that we're facing, such as increased growing season temperatures, reduced and more variable precipitation, spread of new and existing pests and disease, as well as sea level rise, which has in turn had many different types of impacts on agriculture, like reduces in yield, reduced irrigation water availability, as well as increased harvest loss due to pests and degradation and loss of coastal farmlands. But what I think is exciting is that in several cases, there's been adaptive crop wild traits that have been identified to help uh, maybe somewhat mitigate these impacts, such as looking at morning flowering in rice for heat tolerance, or looking at drought tolerance in wild wheat species, or late blight resistance in wild potato, and salt tolerance in wild sunflower. So one thing I'm really excited about is I think that these crop wild relatives have a lot of opportunities and they could be very important for breeding climate resilient crops. And so just because I think everyone has a slightly different definition of what a crop wild relative is, I am going to define it here as a taxon related to a species of direct socioeconomic importance. And so this includes food and fodder and forage crops, medicinal plants, ornamental and forestry species, as well as plants that are used for oils and fibers. So in this image in the left, we have a wild helianthus and then our sunflower crop, some wild beans, as well as wild wheat and wild corn. And there has been proof of concept that has shown how important these crop wild relatives are. So in 2022, a new potato variety for farmers was released, the CIP Matilde is I think how you say it, and it can almost completely withstand late blight. That is really devastation is quite devastating to potato crops. And one of its parents is a wild potato, this Solanum species, which has blight resistance. And this is really the first time that potatoes derived from wild relatives have addressed traits that were asked by the farmers. And this is from Crop Trust. As well as this example that was also in 2022, a new drought-tolerant variety of durum wheat with a pedigree that also includes a crop wild relative was released for cultivation in Morocco. So unlike other durum wheat varieties joining the region, this wild genes enables it to kind of survive in these really tough droughts. This is typically a wheat that's used to produce pasta, but also is important for couscous and bulgur. So the big question is, so how do we figure out which crop wild relative is best for building climate ready crops? And if you look at different definitions of what a crop wild relative is, some ways they're defined as these taxon groups. So a taxon group 1A is a crop itself, 1B is the same species, 2 is the same series or section, and then all the way out to taxon group 5, which is the same tribe but a different genus. And so for some crops, this can be hundreds of species. And so, what I argue is that the herbaria is a really great place to have a first pass to try to understand maybe a place kind of used as a magnoscope of where we should look. There are about 3,600 herbaria worldwide, as well as 400 million specimens. So there's just a ton of data, and this is just an estimate. There's probably way more, especially in smaller herbaria, which are almost more important, but not typically digitized yet. So we're working on it. And the best place on where I work is AgBio, which is this national repository for data and images for millions of biological specimens. And these are available in these electronic formats. So here are these image data for some crop wild relatives. And this is available to anybody, really. And unfortunately, kind of, I know you are all thinking about this too with sustainability. That may change, but at the moment, they're available to everybody. And so those of you that may not be as familiar with AgBio, it stands for Integrated Digitized Bio Collections. And this data is pushed from museum and herbaria, universities, state and federal agencies, as well as community scientists. So all this data is pushed into this repository, and then that's available for really anyone. But today, what I'm going to focus on is some agricultural purposes. And this is really where my role in AgBio stands out. And I'm going to talk about my favorite group, which is Brassica oleracea as a first example of this. I know this group is probably familiar, but this is broccoli, Brussels sprouts, cabbage, cauliflower, kale, kohlrabi, everyone's favorite vegetable crop. But there's about 34 actually different cultivars, including this one on the bottom right, which is actually a walking stick kale from Jersey in the UK. And so during my PhD, I was able to understand the phylogenetic relationships of the cultivars as well as their wild relatives, and kind of better understand the distributions of these species across the Mediterranean. And when I wanted to dive deeper into understanding and growing these plants, the first place I turned to, of course, was Grin. And I was pretty sad, and not the fault of Grin, just as this group knows, it's really hard to find funding and people. And so with the crop wild relatives, there just wasn't many available accessions for these. And so we used what we could in my dissertation, and then more recently have been supplementing it with actually going to collect these plants. But what's very exciting is if you look at the digital herbarium data, it's a ton more data that we can take advantage of to learn from. So it's a good resource that's very underutilized. And one of the ways that we can use this data is take this data, not just from IDIG but also GBIF, and we have these known occurrence records from the herbarium specimens that each have a lat long from where they are, and we can combine it with environmental data. So what I did in this study, in this analysis, was use soil grids as well as World CLIM, and then we can look at the environmental position that a species occupies. So basically the information such as elevation, temperature, precipitation, and soil that is underneath that occurrence record. And when we do that, we can then apply it to things like the minimum temperature tolerance. So, for example, this is Bio6 from World Clim, which is the minimum temperature of the coldest month for these Brassica wild relatives. And you can see there's some variation, not only within a species, but across the species. And then we can compare this to the hardiness zones. I think this min temperature of coldest month is one of the closest variables to kind of match this. And so, for example, if we want to, grow something in this green region across the US, maybe we want to look at Brassica, Montana, especially these kind of outliers, what those collections are, and go do some genomics and genetics to see what's going on there. Or if we want to look more in the southeast of the United States to grow some broccoli, we want to maybe look at Brassica villosa or Brassica rupestris. So this is again just from these records that are in the herbaria. But we can take this a step farther as well, and again, take this same data and this time make or build ecological niche models. So these are correlative models of the environmental conditions that meet a species' ecological requirement, and then we can predict the relative suitability of habitat. So here we have our three known occurrences in the red, and then we can see in this grid here, there's a possible existence in that area because they have shared environmental conditions. So they have these darker grids here. And then we can build these models and then we can do really fun things with them. We can project them to different climate scenarios in both space and time. So we can project this E&M to the current time and current distribution where that species occurs. But we can then also project that model and look at habitat suitability in our current croplands where we're currently growing. So, one goal, of course, coming from more of natural environments is we want to use less area for cropland. So how can we use our current croplands? And then we can also look in the future. So what will this suitability of habitat for these different wild relatives look like in the future? And so what this looks like in practice, this is Brassica cretica. This is a niche model that's been projected to the current time and space where this species occupies. So it's in Greece. These red dots are the occurrence records that were used to build this model. And then the darker blue is high habitat suitability, and the lower or lighter blue colors are low habitat suitability. So as you can see, it's mostly found in Crete. The name comes from there, and this species mostly occurs where there's high habitat suitability as expected. So we can kind of verify our model is doing okay. But we can get other things out of these models, like looking at what of these variables that we put into our model are most important to determine that that species has high habitat suitability. So for Brassica cretica, we found that isothermality contributed about 50% to the model. And so here, how you read this graph is on the y-axis is a predicted value. So 0, you're not going to see that species. 1, you're very likely to see that species have high habitat suitability. And then the value of isothermality on the x-axis. So as you increase your isothermality, you're a lot less likely to see this species. So this is a measure of how the day to night temperatures oscillate relative to the summer to winter oscillations. So it doesn't like a lot of change, likes warm environments. But then we can then take this model and now project it to our croplands where we're growing our crops is kind of broadly. And when we look at Brassica cretica, which in the phylogenetic analysis is a sister taxon to our cultivars, it has basically this really light color. You probably can't see it at all across our croplands. So we have these little red dots. Those are our occurrence records, kind of that zoomed in version we just looked at. But it basically has no suitability in these croplands, or very, very low suitability. But we can look at other crop wild relatives. So there's Brassica cretica. We can look at Brassica incana, and here you kind of see a little bit more of this dark blue popping out. Here, Brassica insularis, even more dark blue popping up. And then here, Brassica villosa, kind of this middle blue, but maybe darker blue in Australia. So maybe there's a different crop wild relative that might be best if we're thinking about trying to learn about how crop wild relatives might make growing brassica crops easier in our current croplands. But then, like we said, we can also take these models and project them into the future. So there are hundreds of models from groups all around the world. There are many different time periods that we can choose from. And then there's also these shared socioeconomic pathways or SSPs that basically kind of say, how well are we doing at mitigating effects of climate change? I'm going to say that too. And so here is what it looks like for these different SSPs. So this is the year and the CO2. So this SSP5 is really bad. SSP3 is kind of middle. And then the SSP2 is around that 3C ideal, and below SSP1 is below the 2C value that was hoped for. And you can kind of see there's these challenges for adaptation as well as mitigation. So you're kind of looking at these balances of what's going to happen. And so for these analysis, we focused on 2 models from 2 time periods for 3 different SSPs. And so this is a little bit hard to read, so I'm going to walk everyone through. But here's our 2 models, our 2 time periods, and our 3 SSPs. And then this is our crop wild relatives again. And then in parentheses is the percent of suitable habitat in current croplands. So you can see, Brassica macrocarpa, which is actually a species that only occurs literally on like one hillside in one island off the coast of Sicily. So it has 0% suitability in current cropland. So you can see what it'll come up as NA for the future. But Brassica cretica, as we saw, very, very small amount of current habitat suitability. So we can ask what happens in the future. Are there winners? Are there losers? And so if we look at Brassica villosa, which has about 20% suitability, it loses across all scenarios the percent of suitable habitat in the croplands, but just kind of a little bit. And then when we look at this for all the species, we can actually see Brassica cretica does gain some in some of these models. But we can look at maybe something more middle of the road and zoom in on things. So we can look at Brassica insularis that currently has around 27% suitable habitat and gains a little bit in this access model and just loses a little bit in this GIS model. So kind of maybe taking a conservative space. And then we can zoom into places where it gains. So we're interested in growing some brassica crops in North America. It gains suitable habitat in northern US and Canada, but also this dark blue color remains unchanged. So it kind of remains suitable. It doesn't go unsuitable. It's not lost in the future. So maybe this is the best crop wild relative to do further analyses with if you're interested in trying to grow in this area. And we didn't just do brassica. I love brassicas, so I always talk about brassicas, but we actually did 16 different crops and for a number of crop wild relatives. And how I determined the crop wild relatives was anything that had a really nice published phylogeny. And I focused on vegetable crops because they're the third most economically important group, but are generally understudied. And it's just one of my favorites. But I also will talk about a quick example in onions, garlics, and leeks. So here is that same setup with our two models, our two time periods, and our three SSPs with our crop wild relatives that we studied and their current amount of habitat suitability in croplands. And then we have a wide array of winners and losers. For this example, Allium rude has about 50% suitable habitat in croplands and gains across most models. And then we can look at where some of these gains are. And for this example, there's a lot of gains in the Western Cape for allium. And then, so kind of overall, what we hope to prove with this study is that Herbaria can provide important insights for developing resilient crop. It's kind of just showing where to look, where to follow up on some analyses. There's genetic variation within and between crop wild relatives that may provide valuable traits for adapting crops to changing environmental conditions. And the most suitable species for developing these climate resilient crops may not be the sister species that are in these phylogenies. And then ecological niche models can help identify the most suitable crop wild relatives for specific regions under projected climatic pressures. So I hope I've convinced you in this first part that these insights are, all derived from the herbarium records, highlighting their really critical role in forming crop breeding strategies. Typically, these are not two entities that talk together very frequently, but I think there's a lot of potential future opportunities to do so. Okay, so this is always fun to talk about these, you know, great research projects we can do. But one thing I've heard is also going on with AgBioData is how do we keep it, sustainable? How do we keep this data safe and this organization, this group that provides this community to help foster this type of research? And so I was asked to kind of talk a little bit about how we're thinking about this at AgBio. And this is our current organization of AgBio. So we have these 6 different domains from administration, digitization, cyber infrastructure, research, our Symbiosis support hub, and our education and outreach domain. And it's been a 15-year project, so it has been going on, and we hope that it will continue. There's been a ton of specimens that have been aggregated, millions, 150 plus millions. There are millions of media records that have been mobilized. There is over 1,000 collections and over 400 institutions that have been engaged. There have been 9 conferences that bring together not only the curators from the collections, but the folks using this data downstream, and then thousands of people that have participated in both these workshops, these conferences, as well as different summits and webinars. We have partnerships that are helping to increase the impact. And I think there has been already, as for you all too, this lasting impact of uh, building this community. But going future, kind of thinking about how we're going to sustain, I think really the big thing is things are a little bit more separate for right now, but there are several things that kind of individuals are pulling and taking forward. I say pulling with them really trying to keep it going. So we may not have like an administration team, but we still have our 5 other domains. So I'll talk a little bit about what they're doing. So, Austin Mayos at Florida State University is really taking on this digitization academy. And so this is the website. So you can go to the website and they have this whole course to really help folks get their collections online. Um, and they have been very successful so far since 2021, have engaged 615 people from 51 countries in 25 courses. And then just recently, they've added a few new ones, this digital imaging for biodiversity collections, as well as this AI in biodiversity collections. And then what is exciting is that in April, they offered a Spanish version of this class that had 270 applications. So there are so much interest in getting these collections from this part in Latin America online, which is hopeful that we can continue to do this. And Austin is really leading the charge there. The data, that's probably the biggest change in the sustainability model for AgBio is no longer being hosted by AgBio itself. It's actually now a node on GBIF. So this is agbio.gbif.us. And so most of the collections are there. There's about 1.7 million that are archived in a separate place that still can be accessed. But that's probably the big difference is that having this data being still accessible just in a new place. Then being led by Jose Freitas at UF. They've also been, this is another part that's going to kind of he's going to continue working on is this iChat bot. So this is basically a conversational interface for this biodiversity data. And it is a community-driven research platform. So there's these services from the Biodiversity Heritage Library globally, IDBio, iNaturalist, GBIF, and they're adding more every day. And so this is coming in 2026 with a supervised orchestration. And so, for example, you could ask, where can I find some species? And then it's going to search all these places and give you some information. Pam Soltis is really leading the research domain. So this is where I am a postdoc, as well as with the USDA work. And so we are still using this data for various types of questions, and then also are working to continue the Digital Data Conference. So this is an annual conference to try to foster and keep that community somewhere that everyone can still meet. It was online this last year as a virtual event. And we've done that a few times, especially with COVID, but hopefully can go back to doing a in-person meet. And then the Symbiotic Support Hub is being led by Al Gilbert out of KU. And this is the current distribution of the US-based collections in Symbiota portals. So they have a really nice range. And so he's working on continuing that connection. So that's really that interface with the collections and getting the data online. And then Libby Elwood is leading the charge with our education and outreach. So there's an iDigBio Events Hub. Here is the information there. So she's leading the charge. There's also a YouTube channel that Libby's continuing. So there's a couple of different ways to continue those workshops as well. So really, these are kind of 5 domains that are continuing forward. And we're hoping that maybe we'll have another 15 years to with this model. But I do want to make sure I acknowledge all the folks that helped with the ecological niche modeling, as well as several of these LASPER folks that are undergraduates. So it's Tori Ford, Jonathan Braz, Sebastian, and Tyler. And would like the funding, and I'm happy to talk a little bit more about anything that folks might have a question about. Thanks.
That was absolutely fabulous. Thank you so much for that really, really interesting talk and
Yeah.
very, very clearly laid out for people to follow. So, are there questions from the audience that people want to? You can. There's a small group of us here, so if you want to just sort of unmute yourself and ask away, give
Yeah,
people
I
a
could
chance.
I
Yeah,
could
go ahead,
start.
Andrew. Mhm.
Thanks for a great talk. Very interesting. I was curious. So the examples of ecological niche modeling that you showed kind of focused on what I would think of as like abiotic stress.
Mhm. Yep.
What are the challenges with dealing with biotic types of issues?
Yeah, that's a great question. Yeah, integrating the biotic information is like the dream. Like gold standard would be great. You got like a full picture of what these species are really responding to, right? To understand why they exist, where they exist. But collecting that data, really hard. So knowing like every organism that they're interacting from pollinators and frequency and it's just not data that is available unless you can collect it yourself for a really small region. So there are some examples where people have done that, but it is really labor intensive. So doing it for like a big study like this, unfortunately, isn't as possible. But I think, what's nice is that for this crop group, right? Like they're being managed. If we're thinking about like what is suitable habitat in croplands that's being managed by people, we kind of can get away with just looking at the abiotic information as this one axis, which we know is, really difficult for the crops to respond to in some cases, in many cases. So there's the gold standard, and then kind of hopefully this is still informative and helpful. Thanks.
Yeah, hi. That was a very interesting aspect of how germplasm or wild species are used. I have a question of just the reverse of Andrew. Are there studies, for example, in insect, the equivalent of Herbaria for us plant scientists, where they predict which would be more prevalent in geographical regions and in future time periods? Are they doing such studies?
That's a good question. So I think that there are some models for insects as well. I'm not as familiar with the insect world, just working mostly in the plants, but there are folks that have been using the collections data of insects to also build models. Of course, the insects, like part of this is defining like where the species can access. And with plants, it's a little easier because they can't go too far. But the insects those can fly, some of them. So it's a little bit harder. But I'm sure that just like I could look online and try to find some examples. I think the question is are they agriculturally relevant examples of insects? So maybe they haven't been done to do them.
Thank you.
Yeah.
Can I follow up with another question? I'm
Yes, and then
curious.
Monica.
Oh, okay. Yeah, after you,
No, no, no,
Monica.
no. Ask your follow up, and then Monica will ask.
Oh, all right. I'm curious about you. You might have already mentioned about the green collections data that you collected. Because for us, when we think of wild relatives, only green comes to mind, never herbarium. Okay. So,
Yeah.
how much overlap is there in all these plant herbarium data, usable data for this purpose, and the green? Are there overlap? Are they collaborating? Or are they very different?
So if I understand that like where what like from the herbarium specimen to the living collections, are they very different?
Um, yes, because they also maintain the passport data of their collections, and you
Yeah.
are sort of using passport data
Yeah,
not for
definitely.
the living but the herbarium collection.
Yeah, I use a lot of the time, I'll use herbarium records when I'm doing field work to try to go collect these to try to get the information of where they were and go visit that site. And many times, not always, they're there still. Yeah, to try to, because then, that's especially variation within the species. It would be really interesting to, collect those ones in that region that would be of interest to understand the genetics. Are there like any more heat tolerant genes in that species, for example, and then going to recollect that taxon.
Okay,
Yeah.
great, thank you.
Yeah.
Uh, Monica?
Yeah, sure. Thanks. I had a question about metadata, and this might reflect sort of my ignorance about plant metadata and the passport metadata that was just mentioned. So apologies if I work with insects. So, so the Symbiota platform is essentially the software where um, both herbaria records and other types of occurrence records can be co-housed.
Yeah.
Okay.
Yeah.
Um,
Any record, any biodiversity from fossil to anything really.
okay, yeah. I was wondering, so since you started from this sort of herbarium standpoint, moving into the AgBio and combination, et cetera, is that sort of changing how people collect plants and what metadata they record when they actually supply their samples to herbaria? Back when I was in grad school, I remember the very basic location level metadata that you
Yeah.
had to have, et cetera, for your plants. But are people actually expanding now and saying, well, I actually took, a pH measurement at that collection site or temperature or whatever, and adding that because the infrastructure to keep that metadata and make it interoperable across within that platform is now available.
That's a good question. I would say I definitely think it's changed how people have collected their data. So, um, like, I think a lot of folks are one of the big things I can say with plants, and maybe I think this might be true with other groups too, is that folks will do like a collection for a herbarium or any natural history repository, but also do like an iNaturalist record, taking a photo. And then, so you kind of have, when you're doing these downstream analysis, now you have these duplicate records and you need to like make sure you can, if you really just want one for models, so you have to be able to kind of tease them apart. But from that information, you can get, from a photo, you can get a ton more information that might not be always labeled. I haven't seen people necessarily do pH. Um, I think it's still pretty much the same thing. I think we're lucky if people write their soil type or the community that the plant is collected with. So this data, this extra data, like the soil and that's all extracted from raster data. So yeah, it would be great if people could be interested, like how much does the metadata collected on the specimen match these layers of data? So I know if you do elevation, for example, which is commonly recorded, that those sometimes don't always agree. So I'm sure that would be true for these other types of data that would be collected. But I think the big thing I see is this like an INAT record, or sometimes people won't even do the specimen anymore. They just do an INAT record, high naturalist. So but that's not ideal, I would say.
All right, thanks.
Yeah, thanks. Matt?
Uh,
Oh, yeah.
Yeah, I'm curious if you've done modeling beyond IPCC's SSP5 pathway, considering it's pretty clear those were a bit too conservative for where we're going.
I know, right? Isn't that just I have not, mostly because those like data are not, at least I haven't been able to find a great repository that has it at the right resolution for the global space. But if you know of one, I'm happy to hear it. But yeah, that's the problem. These are all now almost obsolete. So that
Right.
is a scary reality. So then you want to see like, is one obviously there could be a threshold, right? These ones that look like they're doing well with these increased temperatures or less precipitation, whatever it may be, may hit a threshold and whatever we get to may be too much for them to tolerate too, but then even more so focusing on one that seems to be doing well or, is in a warm region and then this area you want to grow the broccoli is now going to become more similar to that place now that's currently already warm. Yeah.
Right, because at least based on my understanding, like beyond just climatic considerations, like the farther north you go, then you have photoperiod problems as well. So it's not
Yeah.
purely a question of, just minimum and maximum temperature variation. So
No, yeah, exactly. Yeah, this is definitely not like this will work 100% species, but just probably just like a space to kind of start that might be a potential place to look.
yeah,
Yeah.
thank you.
Yeah.
I'm going to sort of throw something out that's maybe more of a comment than necessarily a question, because it makes me think a lot again about, what happens when resources kind of disappear, right? And wondering, you know, what this is doing in terms of revealing the gaps and whether or not there have been resources like that there are some archives somewhere that because of lack of funding or whatever, have sort of, languished on somebody's hard drive
Yeah.
or something. And I'm not sure how to go about sort of uncovering those except through maybe some kind of like, deep historical literature search or something like that to see if, it's possible to resurface those. But it also sounds like you're really identifying important needs in terms of like data collection. Like we need to have this data. And I wonder about the Venn diagram between the data that it's important to collect and the goes against the current policies for data that we should be collecting because it has to do with environment. Like, what are
Yeah.
we going to
Yeah.
lose by not doing this?
Yeah, it is a great comment too. Like so the other way that this data we've like you can use these models is look at kind of gaps. So where we know germplasm for many things for brassica, we have like lat long where that germplasm was collected, and we can look at the overlaps of, we predict this to be suitable habitat currently for this species, but we have in our germplasm only, these areas collected. So then can we use that map to, argue for funding to go do field work to try to collect more of the range that this species should be in so that we can protect things for the future. Yeah,
Matt
thank
just had
you.
a comment, yeah, for you.
Cool. Awesome. Yes, thanks. I will check
Are there
that
any
out.
other? So Olivia has a question. Are crop models like yours readily accessible by growers to breed their own crops using wild relatives as reservoirs?
Yeah, hopefully they will or for crops themselves. It's a little trickier to do because those um you could do for the the crop spaces, but the soils are being modified. So like we need some information of what those soils are actually doing. The climate you could do, right? So probably, but we know plants, right? It's so important with the soils as well, almost sometimes more. So um, I have seen some folks try to do crop models themselves, but I feel, yeah, it's hard to get the information to understand the environment that the crop is responding to because it's being modified by the farmers themselves.
But I
Yeah.
bet that data is being collected. Sorry.
Oh, yeah, it probably is. Yeah.
I have a
Just
question. So I think the digitization of the herbarium is a great thing, and I thought if there is a way to connect and use the phenotype and phenotype ontology and perhaps connect to the genetic databases and genetic diversity in that way. So that would be
Yeah.
something actually we people of the genomics and genetics would greatly benefit.
Yeah, that's like a great point that is a really active area of research right now is this idea of the extended specimen. Have you all come across that?
I mean, like one of the challenges that we have as a genetics and genomics and breeding database, that image storage is a huge space, right? And we often don't have that infrastructure to support the images. And when we talk about phenotype, and if there is that phenotype somewhere in the herbarium and we could link, so then it will be just the hyperlinking, right?
Yeah,
Yeah,
so
so
this
we don't have to build another resource on top of it to have access that image, and I think it will have so much of use for the education, but also for the researchers. And already, a lot of specimens are in herbarium, so that will be something nice
yeah,
potential collaboration.
Yeah, this idea of the extended specimen is exactly that. And it sounds very simple to link things, but what we've learned trying to build even just an example extended specimen, doing that actual linking is really quite difficult. One, a lot of times these URLs or DOIs are not always persistent, especially URLs. And so if you use a URL, in 5 years or 2 years or 6 months, it's gone. So that link is no longer functional. So how we link these, like even just a single link, like here's a specimen and it was in this genetic study. So here's the NCBI accession and here's who collected it. Those links we've been trying to do, and it's usually incredibly labor intensive. Not yet is there an AI model that has been able to tackle it. And then so it's important. I think that would make everything. And then like there's all the phenotypic data, like someone has measured these leaves or has done a photosynthesis analysis on that specimen and has information on that. How do we link to all of those? And it sounds like it should be simple, but there's folks trying it and it has not been able to be done in practice well yet.
So in the reactome and potentially in plant reactome, we are thinking that each pathway can have its DOI ID, and
Mhm.
then people, for example, the students and other community curators who contribute to that DOI, also links their name and they can cite it as a
Yeah,
product.
I think
Perhaps in the herbarium, if a curated specimen with all the information about the plant structure, phenotype, traits, something like that. Perhaps creating a DOI would be a way to do that.
yeah, so there's some that have done that. If you download like a data set from GBIF, you can get a DOI that's unique to that data set that was downloaded. Specimens have like accession numbers. That's the typical format, or barcodes. But like an accession number from California Academy of Sciences or the Chinese Academy of Sciences are CAS 12345 and CAS 12345. Or if you're an insect and you're at the California Academy of Sciences and you have the session CAS 12345 and you're a plant that has in the same building but a different collection, you have the same accession. So how do you know what you're talking about? And so these, yeah, it really is surprisingly really challenging for at least this
How
side of
would it
things.
work? Like, would it work like if people like, for example, Plantriactome or Tier or, some of our MagiDB or something, we collaborate directly with one herbarium and then have somehow those stable IDs or, the pictures or something
Yeah, I think that is, I
directly
think like
linked to it?
any starting place is a good place, right? So for example, I grew up all these Brassica oleracea cultivars during my PhD in the greenhouse. And then I was like, because I come from like a systematics background, I'm like, okay, I need to put my specimen in the herbarium. And then the herbarium curator is like, I don't want your plants that are grown in a greenhouse. That's not a natural state. So no, I won't take your specimens. So like we got to think about something. But then, like, could I connect, right, the germplasm accession to my, and when I did, when I put my genetic data on NCBI, the germplasm information is there, but it's not like linked really. It's just somewhat searchable. But you have to know it's there to go find it, right? And then, so I don't know. Yes, I hope we can figure out a way, but I don't have the answer
Maybe
of how to do it yet.
we could do a small set
Yeah.
around like, one plant and the couple of phenotype. Maybe brassica could be the good one.
Yeah.
Since
Yeah.
you already have that images
Yeah.
and link to maybe one pathway.
Yeah, we have the Brassica DB. Um, and like there's phylogenies that can be linked that are used as specimens and for there's an unlimited. It snowballs really quickly, but it's fun. Yeah, this bioscience article is a good one.
Thank you. So we'll follow up with you at some point.
Yeah, that would be fun.
Thank you so much.
Yeah.
Lots of ideas. Are there other questions from, because I do have one sort of like question related to the sustainability thing. And
That's a
one of
really hard one.
yeah, it is a really, really hard time. So what I'm sort of seeing from for you guys in the in the iDig Bio space is sort of distributing the different kind of components. So instead of like, now we're all under this RCN umbrella, is that different, as you said, different hubs have sort of been able to take and presumably like find their own funding to continue to pursue those goals, so that you have specific funding around the education initiative, conference funding and so forth. So there isn't necessarily the administrative core anymore.
Yeah.
But so how do you keep this sort of connection still kind of going? How did you fill the gap in the administrative core?
So we
Just
haven't done this
passion?
yet, right? Yeah, we're in the same thing, like no cost extension year. So the plan right now is through August. Not so about a year right now, or a year right now. Um, and then so this is kind of where things are going at the moment. But like for that iChat bio, for example, that is probably going to end up really like a PhD project in Jose Fuentes's lab, or postdoc kind of project. So that's where that would kind of continue. And then yeah, instead of applying for like a grant that would cover everybody, like yeah, applying for education grants, that would be more focused on these webinars. The research grants with Pam and Doug focusing on more of the research, but maybe still being able to tie into education and broader impacts, that type of thing. Um, I, yeah, I think that this is what we're working towards somewhat right now, but isn't what we want. Like we would want to keep all of our administration to keep everything going. Like those people, I mean, I'm sure you guys understand it's like impossible. Those folks are the heart and key to keep things, they have all the connections that are necessary. So I wouldn't say this is the ideal situation, but this is currently what may be the only option for a lot of it. And like the data like no longer hosting it ourselves in GBIF. That's at least the data is safe and in good hands, which is really nice. But trying to meet the community together as much as possible. So it's great that we could have this research, this conference, but like the person who usually organizes it is more in this administration team. So what happens to her without her, it's really going to be like a volunteer on a faculty to organize this whole conference and maybe more Zoom features like this. So not quite as um I don't know, I think that maybe not as beneficial as like a full in-person type of conference would be. And it may just be like that for a couple of years, and then we can get past this awful chapter, and then we can hopefully correct.
I hear that. Thank you very much.
Yeah. Yeah.
Definitely,
Thank you.
definitely feel that.
Yeah. Kind
Shushma,
of
did you have
like,
a? Mhm.
yeah, trying to survive through this hard time.
Yep, exactly. Well, it sounds like you guys have really, thought a lot about that. I did have a question too about like the sort of like moving things to GBIF. Was that like sort of like in a plan, sort of all along? Like, was that your sort of worst case scenario? And then, how difficult was it to make that transition?
So luckily, because iDigBio and GBIF actually started at this, or iDigBio started first before GBIF, and GBIF just was the government cared and they invested really heavily in GBIF and like have full-time scientists and a full-time team, and the European Union is bought in. So it's very well supported. So as opposed to AgBio, that was really, some graduate students and a few employees that are running the whole data ingestion. So I think that it was probably always going to end up this way, but I think there was an attempt to try to keep it together as like AgBio as it is right now, and that just unfortunately isn't going to be the possible. So I don't know. It's kind of both answers.
Got it. All right. If there aren't any other questions, I wanted to thank you, Makenzie, for just an absolutely fabulous talk. And there was a request, if it's okay, for like a copy of the slides. If you want to put something like it just as a PDF, we'll post the video and the PDF on the website, or Marcella can take care of that when she gets back. Again, thank you so much. That was really, really super exciting. So
Yeah, thanks. Thanks for having me. Yeah, and it's a great yeah, I always am excited to see more of these like ag data portals. So I like soy KB at Mizzou obviously was a big one that I always learned about, and yeah, May. So it was very, very always exciting to meet folks in charge of these very important repositories and data sets on that side.
thank you. Thank you very
Yeah.
much. Okay, I'm
Yeah,
going to
nice
turn
to
off
meet you
the
all.
recording.